Hello friends, today we will learn about culture media. What is the we will know about introduction and methods of culture media. So, let's begin. Culture media are required to grow the organism from infected material to identify the causative agent. The basic constituents of culture media are water, electrolyte, peptone, meat, meat extract, blood or serum, agar, like this. Let's know about water. First is water, source of hydrogen and oxygen. Water is the source of hydrogen and oxygen. Electrolyte, sodium chloride or other electrolytes. Peptone, it is a complex mixture of partially digested proteins. It is obtained from lean meat or other protein materials such as heart muscle, casein or fibrin, usually by digestion with proteolytic enzymes. It contains proteases, amino acids, peptides, phosphates, amino acids, polypeptides, phosphates, minerals, K and Mg, and accessory growth factors like nicotinic acid, riboflavin. Special brands of peptone such as neopeptone, proteose peptone are available for special uses. Now let's know about meat extract. It is available commercially as Lab Lemco. It contains protein degradation products, inorganic salts, carbohydrates, and growth factors. Now let's know about blood or serum. These are used for enriching culture media. Usually 5 to 10% defibrinated sheep blood is used. In certain media, serum is used. Now let's know about agar. It is prepared from seaweed, that is algae, Gillardium species. It contains mainly long chain polysaccharide, a small amount of protein like material, and a variety of inorganic salts. It is available either in long thread or powder form. It is used in concentration of 2 to 3% and it melts at 98 degrees centigrade and usually solidifies at 42 degrees centigrade. Agar does not provide any nutrition to the bacteria but acts as a solidifying agent only. New Zealand agar has twice the gellifying capacity than that of Japanese agar. Now let's know about types of media. Media are classified in many ways based on physical state, liquid media, semi-solid media and solid media on the basis of presence of molecular oxygen and reducing substances in the media, that is aerobic media and anaerobic media. Based on nutritional factors, that is simple media, complex media, synthetic media. Under synthetic media there are different types. First is enriched media, enrichment media, selective media, different media, differential media and indicator media, transport media, sugar media. Now let's start with simple media. Nutrient broth is an example of simple medium. It contains peptone water and meat extract 1%. When glucose 0.5% is added to nutrient broth, it becomes glucose broth. Peptone water and glucose broth are now we will discuss. When 2 to 3% agar is added to nutrient broth, it becomes nutrient agar. This is this is the simplest and routinely employed medium in laboratory for diagnostic purposes. If concentration of agar is reduced to 0.2 to 0.4%, semi-solid medium is obtained which enables motile bacteria to spread. Now let's know about complex media. All media other than simple media are complex. Complex media have added ingredients for bringing out certain properties or providing special nutrients required for the growth of the bacterium in question. Now let's know about synthetic media. These are prepared from pure chemicals and the exact composition of the medium is known. These are used for special studies such as metabolic requirements. Dubose medium with twin 80 is one example of synthetic medium. Now let's know about special media. First is enriched media. When the basal medium is added with some nutrients such as blood, serum or egg, it is called enriched medium. Some examples of enriched media are blood agar. Blood is added as nutrient agar. Added with the nutrient agar. It may be used for growing number of bacteria, but one specific examination is streptococcus. Example is streptococcus, which requires blood for growth. Lopher serum slope. The serum is added for enriched the medium. The medium is used for grouping Coryn bacterium diphtheria. These media are employed to grow organisms which are more exacting in their nutritional needs. Enrichment media. Some substances are incorporated in the liquid medium which have a stimulating effect on the bacteria to be grown in or inhibit its competitors. This results in an absolute increase in the number of wanted bacteria related to other bacteria. Such media are called enrichment media. These are very useful for culture of fishes where the non-pathogenic or commercial bacteria tend to overgrow the pathogenic ones. Example, Salmonella being overgrown by Escherichia coli. Some examples of enrichment media are tetrathionate broth. Tetrathionate is added with which inhibits coliforms while allows typhoid paratyphoid bacilli to grow. Selenite F broth. Selenite has similar action as that of tetrathionate in tetrathionate broth. Now, selective media. Selective media contain substances that inhibit all but a few types of bacteria and facilitate the isolation of a particular species. These media are used to isolate a particular bacteria from specimens where mixed bacterial flora is expected. Selective media are solid in contrast to enrichment media which are liquid. Example, deoxycholate colate citrate agar. Deoxycholate citrate agar. In short, DCA. Addition of deoxycholate acts as a selective agent for enteric bacilli, that is Salmonella shigella. Now let's know about BSA. Bile salt agar. Bile salt is a selective agent. It favors the growth of only Vibrio cholera, whereas inhibits the growth of other intestinal organisms. Now let's know about differential media. When a medium contains substances which help to distinguish differing characteristics of bacteria, it is called differential medium. Example, McConkey's medium, which contains peptone, lactose, agar, sodium taurochloride, and neutral red. The lactose fermenters insert LF from pink colored colonies, whereas non-lactose fermenters insert NLF produces colorless or pale colonies. Now let's know about indicator media. This media contains an indicator which changes color when a bacterium grows in the in them. Salmonella typhi grows as black colonies on Wilson and Blair medium containing sulfite. Now McConkey agar is also an indicator medium. Due to fermentation of lactose, there is acidic pH which forms the pink colonies in the presence of neutral red indicator. The blood agar is an enriched medium but bacterializing red blood cells give clearing around their colonies, thus it is also an indicator medium. Therefore, some of these terms like indicator, differential, selective or enriched are interchangeable and more than one term may be applied for a single medium. Now, let's know about transport media. These are used in the case of delicate organisms. Example, gonococci, which may not survive the time taken for transit or may be overgrowth by non-pathogenic bacteria. For transport of specimens to the laboratory, special media are devised and these are termed transport media. Example of transport media are Stewart's transport medium is a non-nutrient soft agar gel containing a reducing agent to prevent oxidation and charcoal to neutralize bacterial inhibitors. It may be used for organisms such as gonococci. Buffered glycerol saline transport medium for enteric bacilli. Now let's know about sugar media. Sugar media helps in identification of bacteria. The term sugar in microbiology denotes any fermentable substance. Glucose, lactose, sucrose and mannitol are routinely employed for fermentation taste. Sugar media contain 1% sugar in peptone water along with an indicator and rates indicator 0.005% acid fusin in 1N-NaOH. A small tube, Durham's tube, is kept inverted in the large sugar tube to detect gas production. The colorless medium turns pink with production of acid by bacteria and gas production is indicated by gas bubbles accumulated in Durham's tube. Certain bacteria are exacting in their growth requirements and need serum for their growth. Example, his serum sugars for pneumococcus. Now, let's know about anaerobic media. These are used for cultivation of anaerobic bacteria. First is cooked meat broth, in short CMB, thioglycolate broth. For more details about this media, we will discuss next. Media for testing special properties. Urea medium is used to test the property urease production. Phenyl pyruvic acid is employed for identification of proteus species. Dehydrated media. Dehydrated media are commercially available. These are simply reconstituted distilled water and sterilized before use. With dehydrated media, the process media making has become simpler and quality more uniform. Finally not finished, we will discuss about methods of culture. In clinical laboratory, bacterial cultures are indicated to first is isolate bacteria in pure culture from the clinical specimens and their identification by various tests. Second is determine antibiotic susceptibility. Third is prepare antigens for serodiagnosis of infective diseases. Fourth is maintain stock cultures. Now let's know about methods of culture. There are various methods. First is stick culture, lawn culture, stroke culture, stab culture, pour plate culture, liquid culture. We will discuss one by one. So let's begin from stick culture, that is surface plating. It is the routine method employed for bacterial isolation in pure culture. A platinum or nichrome ware loop of 2 to 4 internal diameter is used. Due to high cost of platinum, loop for routine laboratory work are made of nichrome wire. This loop is first sterilized in the Bunsen flame by making it red hot and cooled by touching an on inoculated part of the medium. Then a loophole of specimen is smeared onto the surface of a dried plate near the peripheral area. This is named as primary inoculum. From the primary inoculum, it is spread thinly only over the plate by streaking with the loop in parallel lines. The loop is flamed and cooled in between the different set of streaks. It is done to obtain isolated colonies over the final series of the streak. The culture plate is incubated at 37 degrees centigrade for overnight. Confluent growth occurs at the primary inoculum and well separated colonies are obtained on final streaks. Single isolated colonies is the best to study the various properties of bacteria. Now let's know about lawn culture. This type of culture media is employed in antibiotic sensitivity testing, disk diffusion method, and in bacteriophage typing. It, it may be employed for preparation of bacterial antigens and vaccines where a large amount of bacterial growth is required. Lawn cultures are obtained by flooding the surface of the plate with a liquid culture or suspension of the bacterium. Culture plate is kept for a minute and then excess material is poured off. Alternatively, the culture plate may be inoculated by a sterile swab soaked in liquid bacterial culture or suspension. Plate is then incubated at 37 degrees centigrade overnight to obtain bacterial colonies. Now let's know about stroke culture. Stroke culture is done in tubes containing agar slope and is employed for providing a pure growth of the bacterium for slide agglutination and other diagnostic tests. Commonly used agar slope is nutrient agar slope. Now let's know about stab culture. Stab culture is performed by a straight wire charged with culture material, that is bacteria, by puncturing deep inside the agar. This technique is employed to demonstrate gelatin liquefaction, oxygen requirement of the bacterium, and to maintain stock cultures for prevention of bacteria. Now let's know about pour plate culture. Tubes containing 15 ml of agar medium in each are melted and kept to cool in a water bath at 45 to 50 degrees centigrade. The inoculum to be tested is diluted in serial dilutions. 1 ml of each diluted inoculum is added to each tube of molten agar mixed well and contents of the tube poured into a sterile petri dish and allowed to solidify. These plates are incubated at 37 degrees centigrade for overnight. Colonies will be seen throughout the depth of the medium and can be counted using colony counter. Now let's know about the uses. To estimate viable bacterial count in a suspension. Second is to quantitate bacteria in urine cultures. Now let's know about liquid culture. Liquid cultures in test tubes, screw caps, bottles or flask may be inoculated by touching with a charged loop or by adding the inoculum with pipettes or syringes. This type of culture method is adopted for blood culture and for sterility test where the number of bacteria in the inocula are expected to be small. Liquid cultures are preferable for specimens containing antibiotics and other antibacterial substances as these become ineffective by dilution in the medium. Liquid cultures are also preferred when large yields of bacteria are required. Now let's know about the disadvantages. It does not provide a pure culture from mixed inocular. Second is identification of bacteria is not possible. Now let's know about incubation of culture media. Most of the pathogenic organisms grow best at 37 degrees centigrade, that is body temperature of human being, and the inoculated culture media are incubated at 37 degrees centigrade in an incubator. Now let's know about anaerobic culture media methods. Anaerobic bacteria grow only in the absence of oxygen, that is anaerobic conditions. These anaerobic conditions or anaerobiosis can be established by various methods. Methods of anaerobiosis. Let's know about the methods of anaerobiosis. First is production of a vacuum. Second is displacement of oxygen. Third is by displacement and combustion of oxygen. Fourth is absorption of oxygen by chemical and biological methods. Fifth point is by reducing agents anaerobic chamber. Now let's know about production of a vacuum. Cultivation in vacuum was attempted by incubating cultures in a vacuum desiccator, but it proved to be unsatisfactory. This method is not in use now. Now let's know about displacement of oxygen. Displacement of oxygen by inert is gases like hydrogen, nitrogen, carbon dioxide, helium is sometimes employed. Oxygen can never be removed completely by this method. A popular but ineffective method is use of candle. A lighted candle is kept in a large airtight container loaded with inoculated plates. It is expected that burning candle will use up all the oxygen inside before it is extinguished by some amount of oxygen is always left behind. Now let's know about by displacement and combustion of oxygen. Anaerobiosis obtained by McIntosh and Fleiss anaerobic jar is the main reliable and widely used method. Now let's know about procedure. McIntosh and Fleiss anaerobic jar consists of a stout glass or metal jar with a metal lid which can be clamped airtight with the screw. The lid is fitted with two tubes with tabs, one acting as inlet for introduction of gas and the other as the outlet. The lid also contains two terminals which can be connected to an electrical supply. A catalysis is suspended under the lid by stout wire which are connected with the terminals to heat the catalyst for its activity. Nowadays, catalyst at room temperature is used. Culture plates inoculated with specimens are placed inside the anaerobic jar with an indicator. The lid is clamped tight. The outlet tube is connected to a vacuum pump while inlet tube is closed. The air inside is evacuated. The outlet tube is then closed and hydrogen gas is passed through inlet tube till reduced atmospheric pressure is brought to normal atmospheric pressure, which is monitored on the vacuum gauge as zero. Electric terminals are switched onto heat the catalyst, and if room temperature catalyst is used, heating is not required. The catalyst helps to combine hydrogen and residual oxygen to form water. Reduced methylene blue is generally used as indicator of anaerobiosis in the jar. It remains colorless in anaerobic conditions but turns blue on exposure to oxygen. Now let's know about absorption of oxygen by chemical or biological methods. Let's know about chemical methods. Pyrogallol. First point is pyrogallol. Alkaline pyrogallol absorbs oxygen. This method was first introduced by Buchner in 1888. A large tube containing solution of sodium hydroxide and pyrogallic acid placed inside airtight jar produces anaerobic conditions. Now let's know about chromium and sulfuric acid. A mixture of chromium and sulfuric acid is used for producing anaerobiasis. The two chemicals react in the presence of oxygen and produce chromosulfate. Now let's know about gas pack. It is available commercially as a disposable packet containing pellets of sodium borohydride, cobalt chloride, citric acid and sodium bicarbonate. It is now widely used for preparing anaerobic jars. These chemicals generate hydrogen and carbon dioxide when water is added. Hydrogen combines with oxygen in the presence of a catalyst. After the inoculated plates are placed inside an airtight jar, the packet of gas pack with water added is kept inside. The lid is tightly closed. The gas pack technique is simple as compared to filling of gases by cylinders. Now let's know about biological methods. This has been attempted by incubating aerobic organisms along with anaerobic bacteria. Two blood agar plates are taken. One is inoculated with Pseudomonas aeruginosa and the other with specimen of anaerobic bacteria. Two plates are placed one over the other and the sealed along the rims and are incubated. This method is slow and ineffective. Now let's know about by reducing agents. Oxygen in culture media can be reduced by various agents such as glucose, thioglycolate, cooked meat pieces, cysteine, and ascorbic acid. Based on this principle, the two most widely employed anaerobic liquid culture media are thioglycolate broth and cooked meat broth. Now let's know about thioglycolate broth. It contains nutrient broth and 1% thioglycolate. Cooked meat broth. Let's know about cooked meat broth. It is also known as Robertson's cooked meat medium, in short RCM. It contains nutrient broth and pieces of fat-free minced cooked meat of ox heart. Now let's know about principle. Unsaturated fatty acids present in the meat utilize oxygen for auto-oxidation. This reaction is catalyzed by hematin in the meat. Glutathione and cysteine, both are reducing agents present in meat, also utilize oxygen. Third point, sulfhydryl compound also contribute for a reduced oxidation reduction potential, in short OR. Now let's know about the procedure. What is the procedure? Both inoculation, the medium is boiled in water bath at 80 degrees centigrade for 30 minutes to make it oxygen free. For strict anaerobiosis, the surface of CMB medium may be covered with a layer of sterile liquid paraffin. Now let's know about interpretation. Saccharolytic anaerobes, in short CL, example CL porphyrins, turn the color of meat pieces into red while it becomes black in case of proteolytic anaerobes. Now let's know about anaerobic chambers. It is an anaerobic incubation system. It provides oxygen-free environment for inoculating culture media and for their incubation. It is fitted with airtight rubber gloves to insert hands for working with specimens. These anaerobic chambers contain a catalyst, desiccant of hydrogen gas, carbon dioxide gas, nitrogen gas, and an indicator. Now let's know about methods of isolating pure cultures. Surface plating. It is routinely employed in the laboratory. Use of enriched and selective media. It is employed for isolating pathogens from specimens with varied flora. Example, fishes. Third point is pre-treatment of specimen. Suitable bactericidal substances are used for pre-treatment of specimens. Isolate a particular bacteria. Example, particular bacteria concentration and decontamination. Example, contamination and decontamination. Sputum before culture of Mycobacterium tuberculosis. Fourth point is by heating liquid medium. Specimens are heated at 80 degrees centigrade, destroy vegetative forms of bacteria, but spore bearing bacteria survive, and isolation of tetanus bacilli from dust and similar sources. Finally finish the culture media. Any questions regarding this you can ask me in the comment box, and subscribe my channel for more updates, more biotech topics. Thank you.